So thank you for choosing Breakout 1. The topic is understanding and treating CF infections. And the aim is to generate some discussion and some debate. So I'm joined by 4 brilliant speakers who are each going to present for about 5 minutes. Just to let you know, Dr. Laura Nolan is unwell, but we have her slides, and Professor Jane Davies has kindly agreed to talk to a couple of her slides. So that topic will still be addressed within this session. Our speakers have been asked to cover a few things. A progress update, opportunities for future collaboration, the next steps for their research, and why people with CF should care about their research. So I know it was a big ask to do that in 5 minutes, but we're expecting a pretty quick fire round and really looking forward to hearing what you've got to say. There'll be plenty of time for questions and discussion afterwards. So I hope that all sounds okay. So our first speaker is Dr. Darius Armstrong-James from Imperial College London, and he's going to be presenting on TRIFIC SRC, Aspergillus infections. Thanks, Darius.
Thank you very much, and thank you for the invite to talk about our SRC today. So our SRC is targeting immunotherapy in fungal infections and cystic fibrosis. So I guess the name tells you what you need to know. We're basically looking at new immunotherapies for aspergillosis in particular, which is a major problem in cystic fibrosis. And we have this SRC as a collaborative SRC between Exeter, Manchester, Imperial, Brompton Guy's, Nijmegen in the Netherlands, and also the University of Massachusetts in the States. So in terms of the progress of the SRC, this SRC started in late 2019, so obviously there were a lot of difficulties around COVID. One of the major plans is basically to recruit large numbers of patients, as you can see in this schematic, and then undertake immunophenotyping in those, as well as doing mechanistic PhDs downstream of those to look at different pathways that might be targetable for immunotherapy. We've enrolled about 150 patients so far into the study with sputum, blood, PBMCs, etc. The 4 PhD students and the clinical fellow have all got their own pet projects that they've been working through that are now fairly mature actually. And we've had a further internally funded PhD recently funded in April who'll be able to extend the lifetime of the recruitment for the clinical studies for the next 3 years. And we've established lots of biobanking omics pipelines for the clinical studies that we're doing. In terms of future collaboration, we've also had significant additional funding that will speed along our research. So we've had a collaborative award from the Wellcome Trust looking at antifungal drug resistance. And we've had a Nature microbiology paper that was published this year, which tells us about the way that resistance is spreading in cystic fibrosis patients for Aspergillus. So do look that paper up. There's been a further MRC clinical academic partnership for Anna Reid, the lung transplant physician at Harefield, where she's looking at the role of Aspergillus in lung transplant patients, many of whom have cystic fibrosis. So that allows us continuity into those cohorts. Anand Shah will talk about his project, I think, in this session, so you'll hear from him about that. And there's also a Centre for Antimicrobial Optimization we have where we're looking at high throughput phenotypic analysis of Aspergillus, as well as novel diagnostics for fungal disease. In terms of next steps, most of our cohort of students will finish next spring. The various manuscripts in preparation around cytokine therapy, effects of CF modulators on fungal disease, CF cell death modulation, dendritic cell responses, and clinical immunophenotyping in play. The ongoing biobanking that we've discussed. FACS transcriptomic, proteomic and metagenomic pipelines are in place, and we're looking for further funding, which is always the name of the SRC, to underpin those expensive studies, as well as further work on some cell therapies and also Aspergillus ELISpot type diagnostics. So why does this matter? I mean, Aspergillus is still a problem in cystic fibrosis. Many patients have switched to Kaftrio, and that's improving their sputum production, but the serological basis for Aspergillus doesn't seem to be going away, and it'd be very interesting to understand how that plays out over the next few years. There's increasing issues with drug resistance and toxicities and need for new therapies and immunotherapies. And so I'd just like to finally thank the principal investigators, Andy and Alex at Manchester, Adelia at Exeter, Stuart at Queen's, Frank in Nijmegen, Stu at Boston, Pete, Anand, Rosemary and myself, as well as Imogen at Brompton Imperial, and our students who you can see down the bottom. So that's Tom, David, Elliot, Alex, Natalie and Sarah, and also the patients, clinicians and clinical research teams at Brompton, Manchester and Exeter, and obviously also the CF Trust for funding the SRC. Thank you very much.
Thanks very much, Darius. So, Anand, you're up next. I'd like to introduce Dr. Anand Shah, also from Imperial. He's going to present on fungal resistance, evolution and acquisition in chronic lung disease. Thanks.
Thank you. Thanks very much for inviting me to speak. So it's a variation on a theme, clearly. So I'm going to talk about our MRC funded, which was match funded through a VIA scheme through the CF Trust, looking at fungal resistance, evolution and acquisition in chronic lung disease. There we go. So why is this relevant in cystic fibrosis or across chronic lung disease generally? So fungal resistance is really a globally emergent problem over the last 10 to 20 years. It's been kind of well defined, and it really is a One Health problem. So a lot of this is driven by agricultural use of antifungals, which lead to antifungal resistance, which can then be acquired within a human disease if you're immunosusceptible. Now, there's currently, you've heard about antimicrobial pharmaceutical problems in terms of pipeline. Well, antifungals are even more so. There's really only one class of orally available antifungal drug that's used in clinical practice, and that's actually the same class that's used in an agricultural setting, so you can kind of see the problems. In terms of a clinical setting, it really isn't clear, as we heard, what the impact of Kaftrio is going to be on fungal infection and allergy. That data is still to be determined, and people with pre-existing fungal infections obviously are still going to have particularly the cavitatory disease, that's not going to go away. So this is something that's an ongoing problem. And lastly, there's a lot of drug-drug interactions with existing antifungals with CFTR modulators, which makes it even more complicated. So just an update on progress. So this was really set up to be a longitudinal study of sputum over about an 18-month period. So you can see the problems when COVID and Kaftrio hit in terms of sputum production and also the access to samples. Most of this was done remotely, so postal, so we could still carry on the project through COVID, which was absolutely fantastic. But we've managed to recruit 60 individuals, and with a mean sample of 4 sputum samples over the course of that 18-month period. And really, Amelie, who's our PhD student who's been working on this, has done a huge amount of work and processed, around 300 samples, and we've got about over 400 fungal isolates banked, and about 300 of which are Aspergillus fumigatus. So we've sent them across for sequencing with 2 sequence kind of pipelines already established, and we've really kind of done the cross-sectional initial baseline work and now doing the longitudinal samples. And really where we're at now is to do the phylogenomic analysis to see if we can determine from mutations where these mutations are arising, how they're developing over time. Half of these patients are on antifungals, and the overall prevalence of antifungal resistance in this group is around sort of just shy of 20%, which is what's been published already. So it's a significant problem. Alongside that, what's really exciting, we're also doing quite a lot of environmental sampling. So that includes hospital-based environments, but also the patients' homes as well. So we've sent out lots of kind of remote kits for people to start swabbing their home environments with some air samples as well, and also soil samples, to see if we can determine whether they're environmental or nosocomial hotspots, which are driving some of these resistance acquisitions as well. So for opportunity for collaboration, so as you can see through this, we've developed a really very large biobank of fungal isolates from CF and other chronic fungal patients as well, and sputum, which are linked to comprehensive clinical phenotyping. And alongside this, we've also developed biobanks of plasma, urine, PBMCs, DNA, RNA, PAX genes, all linked to the same individuals. And alongside that as well, their home environmental and nosocomial samples. And obviously this also links to a huge sequence data pipeline that we already have. There's a microreact database, which is part of this grant, and also a Wellcome Collaborative grant, which this links into as well, where there's a huge collection of sequence data from well-characterized phenotypic patients. So the next steps, as I said, the primary step really is to complete the phylogenomic analysis, to look at genetic clusters, potential outbreaks, spatial population structures and transmission routes, and pair that with clinically relevant phenotypes. And we're also looking at metagenomic sequencing, both from environmental air samples, but also from the clinical samples as well, to see whether we can use that to actually determine their resistance, as opposed to doing isolate culture and then whole genome sequencing. So that can obviously bring that detection analysis to a much earlier stage. And I think that is it. Oh, no. Yes, that is it. Thank you very much.
Thank you. That's great. Thank you very much, Anand. So next up, I'm going to introduce Professor Martin Welch from the University of Cambridge, and he's going to be presenting an update on the Pseudomonas gas, food and lodging SRC. Thank you.
Thank you, folks. It's a real pleasure to be here, as always. So the gas, food and lodging SRC is essentially looking at trying to understand why microbes, and in particular Pseudomonas aeruginosa, is so at home in the CF airways. What's special about the CF airways that makes this bug and others want to live there. So one thing about the CF airways is that we know that these bugs like to live off the surfactants that are very abundant in the airways. Lung surfactant is primarily comprised of a phospholipid, and just like myself, Pseudomonas likes fats as well in abundance. So we've been focusing in my lab in particular, just to kick off, on fatty acid catabolism. Just to put it into context, I said Pseudomonas loves its fatty acids. Just to put it into context for you, E. coli, probably the best understood organism on the planet, has just one of the homologs of the genes that we're particularly interested in. For example, FADI, it's a dehydrogenase involved in beta oxidation. Pseudomonas has got 20. Okay, that just tells you how much it likes this particular nutrient source. So what Mung in my team has been doing is she's been trying to understand which of those 20 are really important for the bug. She's done a proteomic analysis to look at that. She's identified 2 genes in particular, which we know now play a very important role. She's knocked those genes out and found that the bug survives much less efficiently on long-chain fatty acids. And she's gone and done even better by solving the X-ray crystal structures of those 2 proteins. And we're currently trying to drug those. Elsewhere in the SRC, it's an international SRC. We have components across Europe that are involved. We've been asking lots of different questions, and I can't go into this in any great amount of detail in 4 slides and 5 minutes. But over in Denmark, Pablo Nickel's group, for example, has been asking, where does the carbon go? When it goes in, where does it actually go? And he's been using a combination of various different omic analyses, so proteomics to find out which pathways are expressed in Pseudomonas, metabolomics to find out which metabolites are present, and fluxomics to find out where the carbon is actually flowing inside the cell. And he's integrating all this to produce probably the best model that we will ever have of to date of Pseudomonas metabolism. Why is it important? Well, again, other experiments done in a different lab as part of the consortium in Denmark have been using artificial laboratory evolution experiments to look at why Pseudomonas, for example, evolves to grow very slowly in the CF airways, and which genes are affected and which pathways are affected. And that, again, has pointed to the fact that you can see on the right-hand side of the slide here, metabolism is what it's all about. It's all about metabolism. It's not an inherently malevolent bug. This bug is doing what it's doing in the CF airways because it's a good environment for it. It likes to eat whatever the goodies are that are there. And so there's lots of pathoadaptive mutations that seem to point to that as well. Just in the last couple of slides, metabolism and virulence are very strongly linked. This is work that's been done by Suzanne Heusler's group over in Braunschweig. She's been looking at what metabolites does Pseudomonas really like to eat, and the different variants of Pseudomonas. So she's got over 400 isolates of Pseudomonas aeruginosa. She's genome sequenced them. She's looked at the transcriptome of each of those, and she's looked at the nutritional preferences of each of those 400 isolates. So she's asking the question from the point of view of what's the bug telling us about what's special about the CF environment. In the final slide, well, Marvin Whiteley's lab over in the States has been asking a very pertinent question, that is, in what form does Pseudomonas live in the airways? And it turns out that it lives along with a zoo of other microbes that also co-occupy. I mean, this is a theme that's coming back time and time again today. We're seeing that we have to bear in mind the other bugs that are present in the airways, the fungi, the staph, the Haemophilus, etc. This organism lives in there as part of a larger community. And we're able to recapitulate that very nicely in vitro these days. And just to bang the drum for something which came out of this SRC and is also funded by the CF Trust through a different program, the VIAs, we've recently shown that life in that community of different microbes, life in a polymicrobial environment, radically changes your response to antimicrobial intervention, and not in a necessarily a good way. So I'm going to pause there. I have been asked, obviously, to say what links are there with other SRCs, other research endeavours that are going on that are funded by the Trust. You can see down there there's the Fothergill SRC that was discussed in the earlier session today. is looking at whether we can get a better model for CF in vitro to test new drugs, new interventions, and there's clear links with that here, along with many of the other fantastic bits of research that are being done by the various SRCs. Thank you.
Fantastic. Thank you very much, Martin. So finally, I'd like to introduce Professor Jane Davies from Imperial College. She's presenting an update on personalised approach to Pseudomonas aeruginosa SRC. And you're also covering some of Laura's slides. So you can just stay up here and go straight on through. Thank you.
All right, thanks. Hi, everyone, and thank you very much for inviting me along to talk about this. So PAPA is a personalised approach to Pseudomonas aeruginosa, which arose from SRC funding we got actually in the first ever round that SRC started, and which allowed us to lay the foundation which we've built on in PAPA. The three streams that we have projects around are improving the detection of the organism, so very timely diagnosis with systems that may be more appropriate than what we're using at the moment in the lab. Improving our understanding of the organism, particularly with regard to its virulence, its persistence, and its ability to co-infect, and then trying to look at improvements to therapies. I work also clinically, and we are constantly frustrated by the inability of the antibiotics that we have available to us to actually achieve very much more than suppression in this sort of context. So I'll tell you just briefly about the various work streams. So in terms of infection detection, we've been working closely with clinicians and scientists at the University of Amsterdam for some time now. They're specialists in breath detection, and they have run a number of programs on detecting volatile organic compounds as markers of other types of airways disease. So we've collaborated with them, and we're working on using some of their technologies to try and detect Pseudomonas and probably other organisms as well. This is very adaptable as a technology, but we're focusing on Pseudomonas within this at the moment. And that's led by Anke Hills and Maitland van der Zee in Amsterdam. She works very closely with a breath scientist called Paul Brinkman, who's been closely involved in this too. And then in parallel with that very high-tech approach, we're doing a much more low-tech approach. One of my favourite projects is working with medical detection dogs. So these have been in the news a bit recently because unfortunately the pandemic has meant they've all got rather distracted by trying to smell something more topical than our Pseudomonas. But these are very, very highly trainable dogs who can be trained to sniff out any sort of compound that has a smell, and we all know Pseudomonas does in fact smell. And we've made some quite good progress with a number of these dogs now, training them to very, very specifically smell not only culture supernatants but also sputum samples. And we're moving at the moment onto a type of huff sample that somebody who would not be able to expectorate sputum can produce, and we can present to these dogs. And we hope to have the next round of pilot testing on those clinical samples collected at home by people very frequently in the near future. And John King is our clinical research fellow who's leading on that program at the moment. So if we move on then to talking about understanding, I'm just going to highlight here some of the co-infection and persistence projects. So similar to what you've heard from previous speakers, we're very interested in competition between different species in the airway, and also the ability of different bacteria to persist and apparently live quite harmoniously together. Dom Hughes he was one of our clinical fellows doing a PhD who's just now gone on to a consultant post at paediatrics at King's in CF. And he's got some lovely work out showing how Pseudomonas and Aspergillus interact when they're present together in the same culture, to the extent that with Pseudomonas present in a culture, we're likely to miss Aspergillus actually growing, and that may account for some of our inability to treat these pathogens. Laura Nolan, who should have been speaking after me, is co-supervising Michaela Mossop's PhD with me, and we're also very, very interested in looking at staph and Pseudomonas interactions, and I'll show you a little bit of that from one of her slides. And then in collaboration with a basic science microbiologist at Imperial, who I've worked with for many years, Hugh Williams, we are working with Livia Spiga on her PhD, which is looking at the way Pseudomonas protects itself from being killed within the organelles of innate immune cells. And then thinking about treatment, one of the areas I'd like to flag to you is the types of treatment approaches that could potentially get us into the clinic. And some of these I think looking quite tantalizing. Ronan Murphy's just handed in his PhD, and he was taking a repurposing approach to a drug which is used in a completely different context for multiple sclerosis, showing that it can damage bacterial membranes to the extent that it synergizes really well with conventional antibiotics, and it can shift a resistant organism into being sensitive to those antibiotics, so potentially really pretty useful as an adjunct. And part of the SRC is also the biofilm group at the University of Southampton, led by Jeremy Webb and Gary Connett, who are co-supervising Declan Power, who's using an approach where donor molecules release nitric oxide into an area of biofilm, reduce that biofilm and allow those bugs to be more sensitized to other agents. So just briefly then, collaborations and future opportunities. We've been very lucky at the National Heart and Lung Institute to have Luke Alsop join as a lecturer in microbiology, really strengthening the basic micro aspects of CF research, and very, very, very nicely synergizing with those of us who are more clinical. And his two students, Alice and Luca, are here today. Andy Edwards also at Imperial, and Alain Filou working on various aspects of this with us. But I'm pleased that the SRC has also enabled us to branch out and forge collaborations with a number of other academic links, but a huge number also of people who are interested from perhaps a small biotech or a commercial angle in developing new treatments for CF. And we're working with all of these companies, for either providing them with samples which have got clinical relevance, or else actually testing the molecules and drugs that they may have developed for that. It's been a pleasure to be involved in the origination of the syndicate and the AMR that you heard so much about this morning. And just wanted to flag that all of this collaborative work has been underpinned by the fact that when we started our first SRC, we started collecting and storing every single clinical isolate that was coming from the CF group at the Brompton. Alex Kunanan's here today. She's our current tireless technician who takes all of these and logs them, and we will hopefully contribute quite a lot of these into the syndicate resource, but are also available for other approaches should people wish. So in terms of next step, we really need to leverage some future funding. The SRC's coming towards its end at the moment, this current one. We've got a number of projects. I've been very pleased to hear quite recently that the breath detection project has received some commercial funding. We're going to, for the first time, be giving out home monitors for people to actually use in their homes, and we've got another grant going in for a big fund at the moment. We've got some funding from the Imperial Biomedical Research Centre, and then there's a number of other approaches that we're taking to try and push forward the treatment aspects, including talking to the CF Foundation, who have quite a lot of interest in this space. They have a science conference that some of you may have attended in the past, which we'll be presenting some of this work at next month. In terms of the collaborations and the networks, it's been really great to be very collaborative in this. I was grateful to Jo Fothergill for inviting me to come on to her external experts to advise on PIPE CF, which I think is a really exciting SRC. And we've also been doing some recent work with LifeArc in the space of difficult infections in CF and bronchiectasis. So just to wrap up then, why should people with CF care about this? All of us have a huge amount of bacterial burden, but particularly people with cystic fibrosis. It's antimicrobial resistance in particular is recognized as a major burden, and it's getting worse, not better. People with CF, even if they've got access to highly effective modulators, many of them have damaged airways by the time they start on those drugs. And so chronic infection, I think, is going to persist. We've recently sort of posed this question, and for me, all the answer is yes, we are still going to need new approaches for Pseudomonas and other bacteria into the future. That might be different for a baby born today who gets access to ivacaftor when they're 4 months old. I hope it is, but we've got a long way to go until we see that happening for everyone. But the thing I think is going to be a particular challenge is that people for whom infection detection was previously easy, they'd come to clinic, they'd cough up a sputum sample, they're no longer coughing up those sputum samples, and they're no longer coming to clinic quite so often. So we really do need to adapt so that we don't, by assuming they're healthier, actually miss opportunities to keep them as well as possible. So thank you very much. That's all I'm going to say about PAPA. Thank you to the CF Trust for funding the entire SRC programs. I think it's been a really great initiative, and it's catalysed so much research across UK. I'm going to try and do justice to Laura's slides. I've cut a couple out in the middle, and I would be massively grateful if nobody asks me any questions about science. Luke's sitting there. He probably knows quite a lot more about some of it than I do. So Laura applied for and was fortunate enough to get a very highly competitive so-called Imperial College Research Fellowship, which gives protected time for 4 or 5 years with no teaching, with nothing else really involved, to begin to sort of set up as an independent researcher. Her area of expertise is in Pseudomonas aeruginosa, virulence, competitions, various other aspects of that, and she's very sorry not to be here today. I think we were all hoping that she would. She's just not well. She's got 3 areas of research she would like me, I think, to flag, particularly in case there's anybody who's got any interests in, is that the yellow card for the second talk or the first? Okay, all right, I'm doing badly. 3 areas of research she wants to flag. The first is that the reason Pseudomonas is so successful is it hides itself in biofilms. DNA is a big part of those biofilms, and she has begun to do some really interesting work to understand how that extracellular DNA actually gets produced. It doesn't just sort of come out of the cells by magic. She's got some lovely videos here where you can see in green the fluorescent Pseudomonas rods going into a circular shape and then actually bursting and releasing that extracellular DNA. And this is a mechanism that she's looking into, but seems to involve holins and endolysins, and is obviously going to be quite complex, but could be something that was targetable in terms of reducing the ability of bacteria to form biofilms. She's been interested in other roles of extracellular DNA in persistent infection as well, and has recently produced some very nice work to show, counter to everybody's previous beliefs, that Pseudomonas is capable of taking up extracellular DNA from another organism, so-called natural transformation, and in that fashion could generate further mechanisms for antimicrobial resistance or other strengthening tactics, for example. And then I've mentioned this project already. This is Michaela's PhD, looking at interbacterial competition between Staph aureus and Pseudomonas aeruginosa within the CF lung. First of all, saying there's a lot of conflicting literature over whether mono-infection or dual infection is worse for people, so trying to get a really, really big clinical data set and pin that down. Secondly, then, to have a look at the way molecularly those 2 organisms speak to each other, not in a culture dish, because we've heard already about how that can be quite inappropriate, but actually in a system that more similarly recapitulates what's going on in the CF lung. And then the really translational bit of that is whatever the findings are that come out of that, we're then going to actually go to CF sputum and have a look whether those molecular mechanisms that we think we've identified can actually be seen within the CF sputum. So final slide. There is a fairly big collaborative opportunity for somebody here. These are the outcomes that Michaela has gathered off the CF registry data set that we have for patients with either single or dual infections of those 2 pathogens. And with this, we're able to identify patients who've given samples into the lab. We really want to try and pin this down, which is the infection type that is worse for people. And that type of collaboration, I think, can most successfully be done with somebody who's really expert in model generation and statistical analysis. So if anybody has those skills or knows a friend who does, please either get in touch with Laura or speak to Michaela, who's put your hand up, Michaela. And it would be very nice if we can take that forward. So thank you very much.
Thank you very much, Jane, and please pass on our thanks to Laura for preparing her slides. I hope she feels better soon. So, speakers, would you all like to come back up to the stage? We've got a quarter of an hour for questions from our audience. We should have a roving mic because we are still recording this. So, does anyone have a question, a comment, an observation? Yes, down here at the front. Well done, Jacqueline.
I work in an adult clinic, and we're all beginning to realise what you paediatricians have been dealing with for years, which is no sputum. And I just wanted your advice on what do we do with all these patients. We're not going to bronchoscope them all, so what's the best way forward? And do we see different sputum cultures? Because we've noticed far more gram-positives, much less Pseudomonas, and much more staph in the cultures that we've done post-Kaftrio. And I don't think that's about what's in the lung. I think it's about what the lab's growing. So
I think it has always been difficult. We rely on suboptimal methods like cough swabs, throat swabs. We know that they lack sensitivity and specificity. We have started in our paediatric clinic to do a lot more sputum induction. And it's really, really useful, but it is time consuming and therefore quite expensive. And people can't do it sort of very, very regularly. It just takes up too much time. But I suppose my approach would be surveillance for those who haven't particularly got worried about with the best tools that you've got, and that probably is going to be cough swabs or something. The minute you feel a bit concerned about a person for any clinical reason, then that's when we would sort of usually trigger a sputum induction. And we've cut down massively on our bronchoscopies by doing that, because we quite often find something that we wouldn't otherwise know was there.
Does anyone else want to speak to that, or should we go back to the audience? No? Okay, so we had a question just here. Thank you.
Hi, I've got a question for Martin, if that's all right. I was interested in the carbon sources for your bacteria, and I wondered if you see any differences in the genes that are regulated in CF-related diabetes versus CF, and whether you've even looked at that, because there is a potential source of glucose for those bacteria.
Oh, I like that question. I like you. No, we haven't, is the short answer. But when I told you that Pseudomonas really loves fat, it really loves fat. It doesn't do carbohydrates. So it will live off glucose as a primary carbon source, but it would prefer to live off other things. So its carbohydrate metabolism is wired up quite differently compared with, say, E. coli. So E. coli is a sugar lover, it's got a sweet tooth, whereas Pseudomonas seems to prefer the fatty stuff, and amino acids as well.
But it will utilise it if it's there, will it?
It will utilise it if it's there. I see where you're coming from. I'll give you a gut feeling answer, and that is it's probably not going to be a major driver. Okay?
Thank you. Other questions or comments from the audience? Yes. It's just coming around here.
Just following up on that question with the fatty acids, you mentioned there was 20 different pathways for the metabolism. Are they generally using one pathway, or they're all doing different things?
There are 20 different homologs for that particular gene that I was very interested in there. That's FAD-E, which is a good drug target. Most of those are probably for the metabolism of weird and wacky acyl chains and things that are not necessarily directly linked with what we conventionally think of as long-chain fatty acid catabolism.
Mm-hmm.
Okay.
Okay, thank you. Anyone else? Yes.
Just a question about this fungal infections. Did you study the variation in fungal prevalences in the country? Is it all same in London and in Manchester?
Well, we haven't studied it specifically, but we know that there's both very high levels in Manchester and London, funnily enough, which is where we're focusing. One of our students, Natalie Francis, is looking at that. So she's basically looking at the CF registry more broadly. And in the last 2 years, Natalie did a lot of work to include extra data fields in the registry so that we can record in more detail the prevalence of Aspergillus across the CF community in the UK.
Just to add to that, there's not a lot of standardisation in terms of fungal culture across the different diagnostic laboratories as well. So probably between Manchester and the Brompton, we probably, I suspect, do quite a different approach, which can increase the yield of fungal growth as well. So if you're looking at a UK-wide, I think I know there is an ISHAM group looking at this, but actually standardising the fungal culture is probably the first thing to do, in which you can then give at least a standardised platform in which to then analyse prevalence, because otherwise you're probably going to be biased. Yeah.
Jane.
Can I just add to that? I think you've got actually one of the biggest problems in terms of detection. As people become non-sputum producers, fungus and atypical mycobacteria are going to really be a problem, probably much more so than things like staph and Pseudomonas. So it's a massive challenge.
Yes. Yeah. And we're doing we're developing lots of molecular assays to address that issue.
So I think some of the approaches that you've got, Jane, in terms of exhaled breath condensate, there's very little bit of data showing it might be helpful, but actually getting lower airway samples are probably going to be invaluable.
Okay. Yes, right at the back there. Sorry, Jacqueline. Back corner.
This is more of a sort of general question and to see where the field's moving. I mean, it's quite clear that you've got polymicrobial communities talking to each other and affecting each other. What about the epithelial cells, and have we got any good models where we can look at that interaction? Because clearly the epithelial cells do interact with the microbiome, and how important is that? And do we have any good models of that? Or is that what you're developing?
Anyone?
Doing more of that than I am.
So just to answer on fungal disease specifically, we have a PhD student who's looking at epithelial fungal interactions using CF knockout, and the interaction of CFTR modulators as well in terms of that interaction. So there are models available, and obviously within submerged ALI cultures and dual cell cultures, the models are there within for any pathogen, but we are doing that with Aspergillus. But I imagine that's the same for use as well.
Nearly all the models have limitations. You know, even the really good ALI models, they lack an inflammatory cell component. So at best, we're sort of taking a part of the puzzle and then hope that another part of the puzzle will come together with it in the absence of a live animal CF model, which fortunately at least we do have now. It's just that they're incredibly difficult to get hold of and to do anything other than very, very low throughput because of the rearing and costs associated with that. But otherwise, I think I'd quite like the approach of a number of different models being used to try and patch in, if you like, the various different components. But that is one of the areas of focus for the AMR syndicate, and certainly also for Joe's SRC, is to try and have a look at getting as good as we can at these model systems. I think certainly for the co-infection processes, there's some very good data that the epithelium's really key, and looking at the infections without acknowledging that may just give us half the picture.
Okay, thank you. Anyone else want to add to that? Okay, anyone else have any questions or comments? Yes, please go ahead.
It's a question for the panel. Do you think that with the recent increase in air travel, traditionally, of course, patients haven't traveled that much, if they do so, that increases the risk of acquiring this Pseudomonas, fungus, or any other weird infections? Or do we see much more prevalence in residences around airports, any major airport?
Jane?
Trust you, Anaban. I don't know of it that there is any evidence. I had somebody working with me a few years ago who did a literature search into air pollution and Pseudomonas, and actually, in general, air pollution, there's not very much about it there. In terms of specifics of air travel, I don't know whether there's anything to do with the proximity and the air changes and everything that we're focusing on now because of the pandemic. I've never seen anything. And I would think it is more likely that there are certainly sort of environmental niches where it's more common to get Pseudomonas if you live in that particular climate next to that particular water. So I would think it's probably a small contributor, if anything, but I don't know that there's any data. Be interested to hear if anybody does.
Agree with that from a fungal point of view. I think there's probably other environmental factors. So, we know sort of industrial composters, for example, are likely to be kind of hotspots for Aspergillus. But even that data has not, really been confirmed yet. and really there's no data in terms of air pollution and fungal exposure, something that we're aiming to look at. But yeah, it's not clear.
Yeah, and the UK's got one of the highest prevalences for Aspergillus anyway, so already know the hotspot.
Okay, I've got a question. Can't see any hands. So, is there anything that you've seen or heard today?
It's only a little one.
Go ahead. Go ahead.
Is there anything association-wise with the housing stock? Because we're always told if you're having housework done, then your Aspergillus rates are going to soar. And we've got a notoriously elderly housing stock across many areas. So
yes.
fungal disease.
Definitely the case.
Have you found it yet? Or is this,
Well,
I mean, that's kind of what we're doing at the moment. So we're looking, doing home sampling for that reason, because it's certainly likely to be more of a problem in certain houses, and there's a whole industry on how you deal with that thereafter, which is complicated, I think, to say the least. But there are groups, there's a group at UCL that actually specialise in that in terms of how you can quantify home mould exposure and what that then means for, insurance companies that have to then go in and decide what to do. But it's something that we're looking at.
Okay. Yes.
In terms of the antifungal problem and only effectively having one class, where do you see that going in the future? The resistance does look to be spreading, and obviously this is a concern, but you think it's going to just be a mounting concern, or?
Well, there is actually a sort of quite good antifungal pipeline now, so there are probably 2 or 3 drugs that are likely to come into play in the next 3 years, which are all novel classes, so we hope that those drugs will make it, basically.
And do you think they'll be used in agriculture as well, or they'll be?
We hope not. They're being, developed for medical uses, so hopefully not. But that's obviously one of the areas of stewardship we need to be very careful about.
I think one of the slight frustrations is that a lot of these pharmaceutical compounds, that when they go into phase 3 trials, they're really being used within an invasive fungal setting. So actually chronic fungal disease tends to be an exclusion factor from those trials. So I think there is quite a lot of work that needs to be done in terms of actually building a model so that industry or pharmaceuticals can look at this as a group to target, not just invasive fungal disease, which doesn't actually include any of the patients that we'd be talking about now.
Okay. Well, it's nearly time for the break. So, are there any final questions or comments, or any final remarks that the panel wish to make?
Sorry, I was going to ask a
go ahead.
A half-trivial question. I heard dogs mentioned, and as I like wine, I know that we can actually ourselves smell down to about 6, maybe 2 parts in a trillion for methoxypyrazine. What sort of, have we any idea what the dogs are smelling? I mean, they've come into their own in Parkinson's disease. Do we have any idea what they're smelling and what sort of levels they're down to?
I think the second answer is easier, in that what I've heard quoted is 10 million times more sensitive than our noses. So if we're smelling down to that, I don't think my nose can smell what you've just described. That's probably why I don't mind cheap wine. But but supposedly 10 million times more sensitive in terms of what they can smell. It's a very good question. No, we have no idea what they're smelling, and that's, if people want to criticise this programme, that's what they go for. They say, but you've got no idea what they're smelling. I think we've got no idea what they're smelling when they sniff out a landmine or when they find somebody at a customs unit either, and that doesn't mean that they're not useful in that context. But what I think people do have a hope for is that if we were able to train them to be really, really good at low levels of a particular infection, that we could try and present them with a number of the candidate choices to say, let's see if one of these triggers a bit of a tail wag or something, and we think that we could then build that into an electronic nose or a box of some sort. My gut feeling is actually they might be smelling a pattern and not a molecule or even 2 molecules. So whether we'll ever get there, I don't know.
Great, thank you. That's a nice question to end on. Everyone's faces lit up when the pictures of the dogs came. I just think, note to the Trust for next year, let's have a dog-related session, maybe even the dogs themselves running a breakout. So listen, thank you so much to our 4 speakers for doing a terrific job this afternoon. And thank you to you for your questions. We've now got a 15-minute tea and coffee break just outside the room, so do carry on the conversation, and we'll return here for 3 o'clock for session 4. Thanks, everyone.